Welcome to an exploration of one of the most fascinating crossroads in modern science, where biology meets nanotechnology. Imagine a world where tiny biological molecules can control non-living nanomaterials. This is not science fiction, but cutting-edge reality. Let's start by understanding what microRNAs actually are and why they matter. MicroRNAs are tiny molecules, only about 22 nucleotides long. These small biological regulators control gene expression in our cells. In physical size, 22 nucleotides translates to approximately 7 nanometers in length. These tiny molecules regulate about 60% of all human genes, making them crucial controllers of cellular behavior. They work by binding to messenger RNA molecules and either silencing them or triggering their degradation. Now let's understand nanomaterials and their unique properties. Nanomaterials are structures with at least one dimension, between 1 and 100 nanometers. At this scale, materials behave very differently. 1 nanometer equals 10 to the negative 9 meters. That's incredibly small. For example, gold nanoparticles typically range from 10 to 50 nanometers in diameter. Carbon nanotubes have diameters of only 1 to 2 nanometers, but can be micrometers long. At the nanoscale, surface area becomes inversely proportional to size, giving nanomaterials unique properties. Let's visualize just how small these entities are compared to familiar objects. Here's the scale. MicroRNA at 7 nanometers, typical nanomaterials at 50 nanometers, viruses at 100 nanometers, and cells at 10 micrometers. A typical cell is about 1,400 times larger than a microRNA molecule. Yet these tiny RNAs can control nanomaterial behavior. Let's use an analogy to understand this paradox better. Imagine a sailing ship. The wind outside pushes it forward, representing external physical forces. But inside, a computer controls the rudder, representing internal biological regulation. The wind represents the physical and chemical properties of nanomaterials governed by materials science. The computer code represents microRNA regulation, governed by molecular biology. The ship's behavior is a function of both wind and code. Similarly, nanomaterial behavior becomes a function of both physical properties and biological regulation. Now the key question: how exactly can biological RNA molecules control non-biological nanomaterials? The first mechanism is direct surface binding. RNA molecules can attach to nanomaterial surfaces. Picture a gold nanoparticle with RNA molecules binding to its surface. The RNA creates a biological corona around the particle. The binding is characterized by a dissociation constant Kd, equal to the product of free RNA and nanoparticle concentrations divided by the bound complex concentration. Typical dissociation constant values range from 10 to 100 nanomolar, indicating strong binding. The second mechanism involves modulating how cells take up nanomaterials. MicroRNAs inside cells can regulate proteins that control endocytosis, the process by which cells internalize nanomaterials. The uptake rate is proportional to both receptor concentration and nanoparticle concentration. MicroRNAs control receptor levels. Let's work through a concrete quantitative example using gold nanoparticles. Consider gold nanoparticles with a diameter of 20 nanometers. The volume equals 4/3 pi r cubed, which for a 10 nanometer radius gives us this expression. This works out to 4.19 times 10 to the negative 24 cubic meters per particle. Gold has a density of 300 kilograms per cubic meter. So each particle has a mass of about 8.1 times 10 to the negative 20 kilograms. The surface area equals 4 pi r squared, giving us 1.26 times 10 to the negative 15 square meters. Now, an important question. How many RNA molecules can actually bind to this surface? Each RNA molecule occupies roughly 3 by 7 square nanometers, or 21 square nanometers of surface area. Dividing total surface area by RNA footprint, we get approximately 60 RNA molecules can bind to 1 nanoparticle. What happens when RNA-coated nanoparticles enter cells? They trigger fascinating biological responses. One RNA-coated nanoparticle can release its RNA cargo inside the cell, affecting multiple target genes simultaneously. Each microRNA typically regulates about 200 different messenger RNAs. So 60 RNA molecules from one nanoparticle could potentially affect 12,000 genes. That's enormous amplification. This creates a massive cascade effect, where a single nanomaterial particle triggers widespread cellular changes. There's a deeper paradox here, related to thermodynamics and information. Here's a fundamental question. Can RNA, which is governed by biological rules, truly control inorganic matter governed by physics? The free energy of binding equals enthalpy change minus temperature times entropy change. RNA binding to nanomaterials is thermodynamically favorable with negative free energy change. Typical binding free energies range from negative 35 to negative 45 kilojoules per mole. The interface between biological and physical systems creates emergent properties that neither system exhibits alone. This paradox has profound practical applications, especially in targeted drug delivery. We can design nanoparticles carrying drugs in their core, with RNA molecules coating the surface for targeting. The RNA coating directs the nanoparticles to specific cell types by recognizing cell surface receptors. Targeting efficiency equals the concentration of nanoparticles at the target divided by total nanoparticle concentration. RNA-guided delivery systems can achieve 70 to 90% targeting efficiency, far better than untargeted approaches. Let's calculate typical working concentrations for these systems. A typical therapeutic dose is 1 milligram of nanoparticles per kilogram of body weight. For a 70-kilogram person, that's 70 milligrams total. The number of particles equals the total mass divided by mass per particle. This gives us 8.6 times 10 to the 14 particles, nearly a quadrillion nanoparticles. The average human blood volume is about 5 liters. So the concentration is 1.7 times 10 to the 17 particles per cubic meter of blood. Let's examine the energy involved in these RNA nanomaterial interactions. How much energy is actually released when RNA binds to a nanoparticle? We established that delta G is about negative 40 kilojoules per mole. Per RNA molecule, the energy equals negative 40,000 joules divided by Avogadro's number. This works out to 6.6 times 10 to the negative 20 joules per binding event. For 60 RNA molecules binding to a single nanoparticle, total energy is 4 times 10 to the negative 18 joules. That's tiny but significant at this scale. Compare this to thermal energy kT, which is 4.1 times 10 to the negative 21 joules at room temperature. The binding energy is about 1,000 times thermal energy, making it very stable. The most fascinating aspect is the feedback loop between nanomaterials and cellular responses. Nanoparticles enter cells, trigger RNA production, which then modifies more nanoparticles, creating a feedback loop. The rate of RNA production equals K, 1 times nanoparticle concentration, minus K, 2 times RNA degradation. The rate of active nanoparticle formation equals K, 3 times RNA concentration times nanoparticle concentration. This coupled system can create oscillatory behavior, where nanoparticle activity cycles over time. Understanding this paradox requires integrating multiple scientific frameworks. This field sits at the intersection of biology, materials, science, physics, and chemistry. All 4 disciplines must work together. Complete understanding is a function of biological, materials, physical, and chemical knowledge combined. Let's examine the timescales involved in these interactions. First, consider how quickly RNA binds to nanoparticle surfaces. The binding rate constant is typically 10 to the 6 per molar per second. Binding time equals 1 over K(on) times RNA concentration. At a typical concentration of 100 nanomolar, binding time is about 10 seconds. Very fast. Next, cellular uptake of these nanoparticles. Uptake time is typically 30 to 60 minutes, much slower than binding. Finally, resulting gene expression changes take 2 to 6 hours to manifest, the slowest step in the process. Let's see how this applies to real cancer treatment scenarios. A tumor with RNA-coated nanoparticles, targeting it. The RNA recognizes cancer-specific markers. Consider a tumor of 1 cubic centimeter volume. That's 10 to the negative 6 cubic meters. With cells being roughly 10 to the negative 15 cubic meters each, that's about 1 billion cancer cells. We need to deliver therapeutic nanoparticles to each of these billion cells. At 100 nanoparticles per cell, we need 100 billion nanoparticles total. That's a total mass of 8.1 nanograms, a surprisingly small amount. There's a profound information-theoretic aspect to this paradox. Here's a deep question. Who actually carries the information? The RNA or the nanomaterial? An RNA sequence of 22 nucleotides carries about 44 bits of information. But the nanomaterial structure also encodes information in its shape, size, and surface chemistry. Total system information equals RNA information plus nanoparticle information plus interaction information. The interface between biological and physical systems creates emergent properties that neither system exhibits alone.